Yeah.
Hello, everyone. Thank you for coming back for day two of the bird monitoring symposium. Today's mini symposium is understanding causes of bird population change. And so, like basically, in recent years, we have documented declines in bird populations, and this has become a matter of global concern, but with implications for ecosystems, biodiversity, and human well-being. And while we have started recording these declines, research into the causes of these declines have been lagging. So it have been lagging. So the point of this mini symposium is to bring together some of the various scientists in the country who have actually been looking at long-term population declines and trying to look at the causes of these declines as well. And that is the what we're going to do today. Sorry, I didn't introduce myself. I'm Tarun Menon, and I am a PhD student at the Center for Ecological Sciences. I study bird migrations in the Himalayas, and I also have a very keen interest in understanding the causes of bird population change. I shall now, without further ado, invite our keynote speaker, who is Dr. Umesh Srinivasan. He is an assistant professor at the Center for Ecological Sciences at the Indian Institute of Science and also happens to be my PhD advisor. He has been working in Eagle Nest Wildlife Sanctuary in Arunachal Pradesh for the last 14 years, monitoring bird demographic rates across land use and elevation gradients. One advantage of having such a long-term data set is the ability to infer causes of bird declines if the study is designed cleverly. In this keynote, Umesh is going to tell us about some of the fascinating results from this data set and also the lessons he has learned while trying to establish such a long-term monitoring effort in order to understand population declines. So over to Umesh next.
Thanks, Tarun. Thanks a lot. Can you hear me well?
Yes, I can hear you.
Okay, thank you. Let me just start sharing my screen. And can you see my screen?
Yes, it's visible.
Okay, fine. Well, folks, Tarun, thank you for the introduction and thank you all for being here this morning. Like Tarun was saying, I'm Umesh and I work at the Center for Ecological Sciences at the Indian Institute of Science. And what our team has been doing for the last 14 years has been tracking bird population dynamics in the Eastern Himalayas, in Eagle Nest Wildlife Sanctuary in Arunachal Pradesh, trying to look at how survival rates of birds are changing over time, and then trying to understand perhaps why these survival rates show the kinds of changes that they show. And so that's what I'll be talking about today. Um, of course, this is a long-term monitoring project, and we are right now in our 14th year. And so obviously, this is not a solo effort. Over the last 14 years, we've had a number of organizations and institutions who have been part of this work in terms of either collaborating with us or funding our work. And we're very thankful to the Arunachal Pradesh Forest Department for supporting this work in terms of not just permits, but just their interest in the kind of work that's happening in Eagle Nest, and potentially using this work to inform on-ground conservation measures. The Ministry of Environment, Forest and Climate Change, the Department of Science and Technology, and of course the Indian Institute of Science for supporting this work. Our work is in the field primarily, and without this set of absolutely wonderful 8 people, none of the work that we're doing right now would have been at all possible. So a huge shout out to these folks. They're fantastic mist netters. They can identify pretty much any bird by song or sight, and great cooks, great company. They have the ability to actually turn a pretty basic camp into what feels like home. So thank you very much to all of these people for their warmth and generosity of spirit and support over all these years. Um, so getting into what we're actually studying, we know that in terms of biodiversity and threats to biodiversity, two of the most important impacts that humans have on biodiversity are via climate change as well as the loss and degradation of natural habitats. And species are not exposed to these threats in isolation. It's not like there's a species that's exposed to climate change, but it's not also suffering from habitat degradation. And the reverse is also true, where species lose habitats or they're forced to actually occupy perhaps suboptimal habitats because of habitat degradation or fragmentation. They're also concurrently being affected by the impacts of climate change. So, although until very recently, conservation biology and studies in conservation biology have tended to look at climate change and habitat degradation in isolation, asking, for example, what are the impacts of habitat fragmentation on populations? Or what are the impacts of habitat fragmentation, population dynamics, or bird communities? It's only of late, perhaps the last 10 or 12 years, that people have actually been saying, hold on, actually, there's other things happening here. Climate change can complicate the impacts of habitat degradation. And then asking what are the joint impacts of these two, what are called global change drivers on biodiversity. So that's sort of the motivation behind why we're doing what we do and why we're trying to monitor these species to understand how both of these threats work together, synergize to impact bird populations. This is particularly important and particularly a little bit scary because the Eastern Himalaya, where we work, is actually warming about 3 times as fast as the global average. And species in these tropical mountains tend to be thermally sensitive. They tend to be species that have adapted over evolutionary time to operate or to live or to be adapted to a very, very narrow thermal zone. And so any perturbations in that thermal zone, whether it's warming or whether it's cooling, are likely to affect these species in various ways. And I'll be talking about potential mechanisms through which these species are actually impacted. But because these species here are thermal specialists, they tend to be very sensitive to changes in temperature. It is concerning what might happen to these species with climate change. Not only are these species thermally sensitive, there are also a lot of species. So if you look at a map of the world color coded by the number of bird species that actually are found across the face of the earth, across terrestrial birds across continents, you'll see that pretty much the hotspots that turn up in terms of the number of species, these are the darker colored locations on the world. The 3 locations that really do turn up as being really, really species rich are the Andes and the Amazon in South America, the Eastern Arc Mountains in East Africa, and then the Eastern Himalayas. In fact, the Eastern Himalayas, in terms of sheer bird species richness, are perhaps the second most biodiverse location on the planet. So there are a lot of species, a lot of thermally sensitive species. And so climate change can potentially impact a large proportion of terrestrial bird diversity that's found on Earth. The kind of habitat degradation that we study is something called selective logging. What you're seeing on the right in that photograph is primary forest, so old growth forest, and what you can see on the right is pretty much forest that has an unbroken canopy. You don't see gaps, you don't see the ground, the bare ground, or you don't see the understory through the canopy there on the right side of that image. Whereas on the left side, what you're seeing is a selectively logged forest. Selective logging is this practice by which uh, people go in and they extract from the forest a certain subset of trees in the forest. So it's not clear felling. Clear felling is more common in temperate areas where logging companies go into these forests and actually remove all the trees from those forest stands. In selective logging, a subset of the trees are removed, either because the individual trees are large or because the species are commercially important in terms of wood, good quality wood for furniture, and so on. So what you're seeing on the left is a forest that has been degraded. You're seeing paths going into the forest, you're seeing trails going into the forest, and those trails are for machinery to go to the forest and then remove timber and remove trees from this forest. And you can see that the canopy starts thinning, the canopy becomes more open, you have sunlight coming down into the understory, you have sunlight hitting the ground. And this changes the structure of the forest. It changes the way in which the forest operates in terms, it changes biogeochemical cycles, it changes the abiotic environment in terms of temperature and humidity, and we expect those changes to have actually quite important impacts on birds. When we work in the Eastern Himalaya, this is in Eagle Nest Wildlife Sanctuary. That white border is that you can see is the outline of Eagle Nest Wildlife Sanctuary. So everything within the white border is Eagle Nest Wildlife Sanctuary. This is in northeastern India. You can see the black dot in the inset map of India. This is in West Kameng district of Arunachal Pradesh, and it's a pretty spectacular landscape. There's a large elevational gradient, and within Eagle Nest Wildlife Sanctuary, you can go from about 300, 350 meters above sea level all the way to the ridge line, which is about 3,300 meters above sea level. So you've got about a 2,000-meter elevational gradient in this area. And within Eagle Nest Wildlife Sanctuary, what you've got are patches of forest that have never been disturbed, primary or old growth forest, and then patches of forest that have been selectively logged. This area used to be, and still is actually, very important strategically in terms of the military. And until about 2004, 2006, there was a large human population within the sanctuary, and the road through the sanctuary had to be maintained by the military. And a lot of the logging that has happened, a lot of the timber extraction from these that has happened in these areas has been because of the need for fuel wood to melt tar to actually be able to maintain the road. So a lot of logging has happened because of that. What you can see on the left, and this is cloud forest at 2,000 meters above sea level. What you can see on the left here is primary or old growth forest. You can see that the canopy is more or less closed, very few gaps. The tree density in these primary forests is about 200 trees per hectare. What you're seeing on the right is selectively logged forest, where the tree density comes down about 2.5-fold to about 75 trees per hectare. And you can see on the right that the canopy is opened up. There's far more sunlight coming into the understory. And that leads to the proliferation of vegetation on and close to the ground, which is called the understory. And you can see a lot more bamboo, dense bamboo in the understory in selectively logged forest on the right. Whereas if you look at the left, because the canopy is closed, there's not much vegetation in the understory itself. It's dominated by ferns with bamboo as well, but not as dense as you can see on selectively logged forest. So there is a massive reduction in the canopy cover in selectively logged forest, corresponding with a massive increase in understory vegetation cover, largely through the proliferation of bamboo in the understory. What we've been doing in these selectively logged and degraded forest patches is monitoring bird populations for the last 14 years. And so what you're seeing in that map there are three patches of selectively logged forest outlined in red and three patches of primary forest outlined in green. And so we've got three permanent forest plots in each in primary forest and in selectively logged forest that we have been monitoring over the last 14 years. The area that we've been monitoring, the area that we've been sampling within this larger landscape comes up to 9 hectares in primary and selectively logged forest each. So a total of 18 hectares of forest that we've been looking at. And what you're seeing in those white markers there are actually locations of mist nets. Mist nets are a pretty standard way of monitoring bird populations. I'll talk a little bit about what mist netting is. But what we've got basically, there are a total of 185 mist nets in this landscape across all these 6 plots. Excuse me. And so what we do every year in the early breeding season, which is April and May, is that we go out into our plot and we place these mist nets, about 24 to 26 mist nets in each plot. And those are the locations in those white markers. And we sample the bird populations using these mist nets for 3 consecutive days in each plot. And then once that plot has been sampled, we move on to the next plot, sample again for 3 days, and then move on to further plots. And so it's about 18 days of mist netting overall per year in this landscape in the early breeding season. Of course, people like Tarun have been working on other projects, but I'm only going to be talking about this. So we also do have permanent plots across the elevational gradient. We do sometimes do some mist netting in winter as well, not just the breeding season. But what I'll be talking about is breeding season mist netting, and the breeding season is April and May in this area. So a mist net is what you can see on the left there. It's a pretty invisible net, especially with the vegetation in the background. It's 12 meters long, 2.5 meters high, and you can see these 4 shelves of pouches, which are these sort of darker colored areas here. And like I was saying, these mist nets are pretty much invisible. Birds don't see them. They fly into these mist nets when they're crossing little trails, and they fall into these pouches or shells. And what we do is we extract these birds from these pouches or shells, and we put on numbered aluminium ring onto the tarsus or the leg of these birds, make measurements in terms of things like body mass and wing length, and then we release these birds back into the wild. And so you can see in the middle there rings of different sizes. Obviously, each species, depending on what size it is, gets a differently sized ring. And the idea behind ringing is to enable us to identify individuals, to enable us to track individual histories. Now, people study tigers and they use camera traps and they get images of tigers in these camera traps. Tigers are easy to identify at the individual level because every tiger stripe pattern is unique, pretty much like our fingerprints. But with birds, we can't do that, right? With birds, an individual bird of a particular species looks pretty much the same as an individual bird of another species. And so, with our naked eye, we're unable to identify individuals in these populations. And so, what we do is to ring them so that we can actually identify individuals. And that's the basis for monitoring population dynamics in these populations. On the top right is a Sikkim wedge-billed babbler, and you can see a ring on its leg there, on its right leg. And this is a bird that we captured, we ringed, and then released back into the wild. This photograph taken by one of the many tourists who come to Eagle Nest Wildlife Sanctuary, and you can see that it's out there in its habitat. It's doing whatever a Sikkim wedge-billed babbler does. And then when we go back the next year into the same locations, and we put out nets at the same location, what we get are either new captures of birds, which have not been ringed before, or we get recaptures. And what you're seeing on the bottom right there is a rufous wing falbetta that has been recaptured. So it's been ringed at some point in the past. It's coming to the net. It's been caught, but it's already been ringed. So we either get fresh captures or we get recaptures. And depending on the relative numbers of fresh captures and recaptures, we can actually estimate survival rates in these species. Over the years, we've had about 12,000 captures of a little less than 8,000 individuals. So we bring 8,000 individuals in this landscape, representing 139 species. And these are all species that either forage on or close to the ground. We're not catching any of the species that forage in the canopy because the mist nets are only 2.5 meters high, right? So we don't catch, for example, minivets or hornbills because they're foraging very high. And you can see the diversity here, just in terms of form and function of these species. Very spectacular differences in species here. Mostly insectivorous birds, but the ways in which they forage can be very different. Now, to remind you, we've been sampling at 2,000 meters above sea level. That's mid-elevation cloud forest in this landscape. And what we expect to see for these different species is that there are species that have pretty much low elevational ranges. Every species has an elevational range, and you've got this species like the white-tailed robin, which is found at lower elevations, and just about comes in to 2,000 meters. So when sampling close to the cold edge of its elevational range, the warm edge obviously is low down in the mountain, the cold edge is higher up, where the species, this particular species has its elevational range. And then there are species for which we're sampling at the warm edge of this elevation, of its elevation range. And what we know from some of our other work is that species are actually shifting their ranges upward. This is not migration. Migration is within year phenomenon where species might be occupying higher elevations in summer and then coming down to lower elevations in winter. This is actually a range shift. This is a geographical range shift. So historically, maybe 100 years back, if a species is found between 1,000 meters and 2,000 meters, today, to keep pace with climate change, to keep pace with warming temperatures, this species might need to move up to say 1,300 to 2,300 meters just to be able to find the kind of temperatures that it is historically adapted to. So we're seeing pretty much more species shifting their ranges upwards, excuse me, and shifting their ranges upwards quite fast. These are changes that we've seen in the last 10 or 12 years. And so what's happening with bird communities in primary and log forests? The primary forest is in green and the log forest is in brown. And this is something called an NMDS. It's just basically a visualization of how similar or different bird communities are from each other. And so what you can see here is that the green dots represent the bird community in terms of what species there are in primary forest and what the abundances of those species are in primary forest in different years. And so the bird community in primary forest between 2018 and 2019 are quite similar to each other because in this space they're closer to each other. Whereas the bird community in 2013 in primary forest is very different from the bird community in 2014 in log forest, because in space those bird communities are very farther apart. So the closer those dots are, the more similar bird communities in composition, the farther apart they are, and the more dissimilar. You can see that on average, which are these ellipses over here, the bird community in primary forest is very different from the bird community in log forest. What's happening here is that we're actually seeing that bird communities in log forests are dominated by smaller species, and they're dominated by species that have, on general, lower elevational ranges, that low elevation species. So log forest tends to be dominated by small, low elevation species. And that might be because of the fact that log forest tends to be warmer than primary forest. I'll show you a little bit of the results that we got from those. These are data on survival rates from the last, for an 8-year period. We're in the process of analyzing the data up to this point, up to 2014 right now. But these are data for 15 species over an 8-year period, pretty much all babblers and flycatchers, and a couple of warbler species. And what we expect to see with these upslope range movements is that birds that are high elevation species, they should move up. So the yellow species, which is the yellow-throated palmetto, given that we're sampling at the the warm edge of its elevation range, it should move even higher. It should move away from 2,000 meters. Whereas the white-tailed robin, which is the blue species below, it's moving up towards 2,000 meters. So the yellow-throated fulvita is going away from 2,000 meters. The white-tailed robin is coming up towards 2,000 meters. So what we're expecting is that 2,000 meters is becoming worse and worse in terms of an elevation for the yellow-throated fulvita. And as it moves away, survival rates at 2,000 meters where we sample should decline. Whereas for the white-tailed robin below, as it moves up towards 2,000 meters, that elevation should become more and more favorable for it, and we should see survival increases in primary forest for that species at 2,000 meters above sea level. These are survival trends over time in primary forest for these 15 species. So what you have on the x-axis is time, what you have on the y-axis is survival rate, and survival rate goes between 0 and 1. So if survival rate is 0.6, it means that 60% of the population has survived from one particular year to the next year. That also means that an individual in the population has a 60% chance of surviving from one year to the next. What we're seeing here, and I'm just going to point out a couple of species, are quite different ways in which species survival rates are changing over time. Some species are showing declines in survival rate in primary forest. Other species at the bottom are showing quite drastic increases in survival rate in primary forest, whereas there are some other species here, like the golden-breasted fulvita, which is G, that for which survival rates pretty much are not changing over time. I'm just going to point out a couple of species here. We've got the snowy broad flycatcher, which is B, and you've got its survival rate coming down over these 8 years from about 80% to about 20%. That's a pretty drastic decline in survival rate. This used to be one of the 5 or 6 most common species at this elevation when we had started sampling. Today, the population rates have gone down very, very drastically. We don't capture this snowy broad flycatcher as often as we would earlier. White-tailed robin, which is in the bottom row there, the fourth species marked by N. When we started sampling at 2,000 meters in 2011, there were almost no white-tailed robins in our sampling plots at this elevation. But over time, this low elevation species seems to have shifted its elevation range to where we are sampling, and its survival rates have gone up from pretty much 0 to about 60% in primary forest. And what we expect to see in that low elevation species show increases in survival rates, high elevation species show declines in survival rates, are pretty much what we see in terms of survival trends. Now, this is showing on the x-axis the difference between the midpoint of the elevation range of a species versus where we're sampling at 2,000 meters. But basically what we have is below 2,000 meters are low elevation species, low elevation relative to 2,000 meters, and beyond 2,000 meters are high elevation species relative to 2,000 meters. What's on the y-axis is the slope of survival on time. So this slope is negative. That slope here for M or N would be positive. So if a species survival is increasing over time, that slope is positive. If a species survival is declining over time, that slope is negative. And if the species survival is not changing over time, that slope would be 0. And that's what's on the y-axis, which is the slope of survival on time. So survival increasing above 0, survival declining if it's below 0. And you can see that the high elevation species here are pretty much showing declines in survival over time. Low elevation species are showing increases in survival over time. Species clustered around 2,000 meters, where the midpoint of the species range is at 2,000 meters, they're not showing very great changes in survival rates over time. But this is what we expect with the range shifts, that low elevation species would show increased survival, high elevation species would show declining survival. And we expected that, given that we are seeing these patterns in primary forests, that the patterns for log forests would be even more stark. That the patterns for log forests would be, oh, if a species is declining in survival in primary forest, given that the habitat is altered, we should see even steeper declines in log forests. So the log forest survival trends are in brown over here. But that doesn't seem to be happening. The elevational range of a species does not seem to be related to whether its survival is declining in primary or in log forest. It's related to whether its survival is declining in primary forest, but not in log forest. We scratched our heads quite a bit about this, trying to understand, why do we see these patterns that are very predictable in primary forest, but we see these patterns that in log forest. And so we went out and we measured a bunch of things about this habitat. Uh, Metrics or measures that we think are important in terms of bird survival, in terms of the persistence of populations in primary versus log forest. The top right and the bottom, the top and bottom left are characteristics of the abiotic environment in terms of temperature and humidity in primary forest and in log forest. Again, primary forest is in green, log forest is in brown. What's on the x-axis is time of day. So this is a 24-hour period that we measured temperature using temperature loggers and humidity using humidity loggers. And on the y-axis in the top left is average temperature in Celsius, and on the bottom left is humidity in terms of percentage relative humidity. So if you look at the top left, what you're seeing is that midday, around 1 or 2 p.m., log forest tends to be on average roughly 2 degrees Celsius hotter than primary forest. So not only is climate change making the entire landscape warmer, logging is complicating the impacts of climate change. You've got all this sunlight coming in into log forest, and that's creating warmer conditions. So the average temperature at midday is about 2 Celsius warmer, which is a massive jump in temperatures. But the maximum temperature, which is not shown here, in log forest is about 6 degrees warmer, 6 degrees higher than in primary forest. That is a massive, massive increase. And so you would imagine that populations of birds in primary forest are in cooler areas, in cooler habitats, whereas populations of the same species in log forest are in significantly warmer habitats. 6 degrees Celsius, just imagine the difference between 31 Celsius and 37 Celsius. It's a massive, massive increase in temperature. And that increase in temperature also leads to a decline in humidity. On average, across any time of day, log forest tends to be 20% less humid, so it's hotter and drier than primary forest. And that, of course, we would expect that to have impacts on arthropods, on insects and spiders that these birds are actually feeding on. And again, you've got an NMDS plot on the right side there. This ellipse is indicative of the kind of arthropod community that you find in primary forest. That ellipse in brown is an average indication of the kind of arthropods that you find in log forest. What we're seeing is quite a difference in the arthropod community composition, primary and log. Arthropods communities in primary forest tend to be dominated by species like Hemiptera, which are bugs, arachnids like spiders, and lepidopterans like butterflies and moths. And these are butterfly caterpillar and moths. These are arthropod groups that are very, very important as prey for these insectivorous bird species. On the right, in these log forest patches, you have species like Phasmatopia, stick insects, you've got dipterans like flies. And you do see, and this is at the order level of arthropods, you do see changes in arthropod communities. So the abiotic environment in terms of temperature and humidity changes, prey availability in primary forest is far higher than prey availability in log forest. And so Akshay Bharadwaj, one of the students in our research group, actually did this for his bachelor's thesis, what he did was put out temperature and humidity loggers along with these mist nets. So every mist net had a temperature humidity logger associated with it. And so when we caught a bird species, we could tell what temperature and humidity it was associated with, what temperature and humidity it was using at the time of capture. What we're seeing here on the x-axis is relative humidity, and the y-axis is temperature. And this is the temperature humidity space occupied by this species, the yellow-throated falbetta in primary forest. This is the temperature humidity space occupied by the yellow-throated falbetta in logged forest. So green being primary, log being brown. I can see straight away that the darker the color means that the more intensively used that temperature humidity space is. And you can see straight away here that uh, the space being used by yellow-throated falbetta in primary forest is far moister, it's far more humid than the space used by the yellow-throated falbetta in log forest. And there is some overlap in the space that's used by populations of yellow-throated falbetta in log forest versus populations of yellow-throated falbetta in primary forest. And the question we asked was, does this overlap actually, or the lack of this overlap in the temperature humidity niches of the species actually tell us something about whether these species fare well or do poorly in log forest? And so what we quantified for a number of species is what is the degree of overlap between the temperature humidity spaces uh, in primary and log forest. So if the species was using exactly the same temperature humidity space between primary and log forest, you would see a perfect overlap in spaces. Whereas if a species and if a population in log forest was using a completely different temperature humidity space than a population in primary forest of the same species, this is within species, then you would see a complete disjunction in the temperature humidity space being occupied by populations primary and log forest. It turns out that if you look at niche overlap between primary and log forest on the x-axis, so towards the left, what you've got is very low niche overlaps. Sorry, let's move down. It's very low niche overlaps on the left over here. So 0.6 roughly corresponds to a 60% niche overlap between primary and log forest. So a relatively low niche overlap. 1 roughly corresponds to almost a perfect niche overlap between temperature humidity spaces in between primary and log forest. It turns out that there's a pretty decent correlation between whether a species can find the kind of temperature and humidity niches that it might be adapted to in log forest, or whether it can't. And so species that have very low niche overlaps uh, tend to be doing poorly. Their survival rates are decreasing over time. That's below zero over time. Whereas for species that have actually high overlaps in niches, abiotic niches between primary and log forest, their survival rates might not be changing or might be slightly increasing uh, over time. Now how these species manage to do this, we still need to understand. Is it behaviorally? Is it species saying? Is it species adapting behaviorally in terms of foraging at different times? And if it's too hot at midday to forage, would they forage earlier in the day or later in the day to find the kind of conditions that they're more adapted to? Um, does temperature and humidity actually have a correlation between the amount of prey that's available? Does microhabitat in terms of small locations of primary forests actually change the arthropod availability in primary and log forest? This is something that other people in our research group are working on. Tarun and Kaling Dangan, two PhD students, are actually looking at bird diets and looking at differences in morphology and morphometrics of populations of birds in primary and log forests, and we're just about getting into those results, and we should happen soon. And so what this tends to indicate is that what we need in terms of allowing species and populations to remain resilient to climate change is primary forests at all elevations in that bird species should be able to move up, populations should be able to move up into primary forest and maintain survival rates. And if they're moving up and they hit log forest or agriculture or settlements, then perhaps they're not going to be able to maintain survival rates. Um, and those populations might be less viable than populations that are shifting upwards, populations that are moving their ranges upwards into primary forest. And some of the work that we've been doing actually very hearteningly actually does inform conservation in this area. There is a critically endangered bird species that you can see on the right there, which is called the Bugun. Its known population size is not more than 20 individuals. Its known global range is about 2 square kilometers, and that's entirely within the lands of the Bugun tribe of Arunachal Pradesh. And so, in speaking with the Bugun community and telling us what our results are, we've actually they have put aside a pretty large part of their traditionally owned and managed community lands to protect birds, primarily the bougainvillea, but also a lot of other bird species that people come to see from all across the world. So right now we've actually got in this landscape a community reserve that spans about 1,300 meters at low elevations, all the way up to 3,300 meters to the ridge line. And we're hoping that with climate change, as bird species start shifting their elevation ranges upwards, at least within this protected area, they should be able to find primary forests to occupy as they move higher up and remain to some extent resilient to climate change. So that's a snippet of the work we're doing. Of course, there's a lot more happening in our research group as a whole. But I'll stop here. Thank you very much for your kind invitation to talk about our work on how climate change, forest degradation affect population dynamics in bird communities in the Eastern Himalayas. Thank you.
Thank you, Umesh, for that amazing presentation, and congratulations on being able to carry out this work for so long successfully. I would request any of the participants who are listening in, if they have any questions, to go on to Discord and ask their questions in the thread. We have a couple of questions already, and I'll read them out to you, Umesh, and you can also read them in the chat box here. The first question is from Suhail. Hi, Umesh. In the slide on apparent survival over the years,
Yeah, I can take that.
Species appear to have shifted. Okay, sure, Umesh. Maybe you can just give what the question is and the answer.
Yes, I'll do that. So Sohail has a question, and what he's saying is that in the slide on apparent survival over the years, some species like the black-faced warbler appear to have shifted survival very drastically, and the data arose from primary forests. Right? So habitat is not changing; habitat remains constant. And Swale is guessing that we're attributing the entire effect to climate change. Just wondering whether climate can actually be changing so rapidly as to change survival so massively from point 8 to point 2. In other words, how reasonable are these estimated changes, and is there some way to address this? So yes, we were actually quite surprised that these survival rates, at least the estimated survival rates from these what are called market capture models, are actually changing so drastically. We did not expect things to go from 80% survival rate to 20% survival rate. So what we've been doing is to triangulate using other sources of information to see whether other expected impacts of climate change might actually be playing out in this landscape. One of the things that we're looking at is upslope range shifts. In potentially in response to climate change. And we are seeing very drastic upslope range shifts in response, likely in response to climate change. For example, black-faced warbler is one of the species that has shifted its range quite to quite a large extent. We're also seeing species that in 2011, when we started doing this work, would have been unimaginable where we're working. Things like great Atacated drongo, things like long-tailed broadbill, things like sultan tit, that pretty much have elevational ranges below, 1800, 1700 meters. We would never catch these birds earlier on. Today, they're in the habitat. And this has just happened over a 10 to 12 year period. So we do think, and also one of our students, Krishna Girish, actually looked at eBird data and looked at past bird species elevational distribution versus present elevational distribution, and actually found that from citizen science data, there are signals of these very rapid movements of bird species in terms of elevational shift. So we're trying to supplement the survival rates analyses with other independent sources of data that might be able to give us a better handle on how climate change is impacting bird species. And this is not just in the Eastern Himalaya, in other places in the world as well, especially in the tropics. Plants, birds, arthropods, mammals. This is a meta-analysis that came out a couple of years back, in that actually species are tracking temperatures in tropical mountain ranges almost 100%. So if you expect, for example, a 1 degree rise in temperature to be associated with a 100-meter upslope elevational range shift, species have actually moved 93 meters higher up. So there's a sort of 93% concordance between what you expect from climate change and what you're actually seeing. So that's we're trying not to rely only on the survival rates estimates, but also bring in other sources of data to try and triangulate and see whether these are indeed perhaps largely restricted to climate impacts and climate problems. So Vishal has a question: do population estimates for some of these species like the snowy-browed flycatcher? We don't have population estimates in terms of saying, there are these many individuals in this population per hectare. But what we do have are capture rates across time. So how many individuals do we catch per unit effort? Per the number of nets that we put out and the number of hours that we sample using these nets. What are the number of captures that we have? And we do see that for species that actually show declines in survival, you do see declines in capture rates as well. Survival declines in primary forest tend to be associated with capture declines in capture rates in primary forest. That's not necessarily true for log forest, where you might have declines in survival rates, but you actually see abundance is not changing very highly. I've not talked about that, but we're still trying to figure out why that's happening. But we do see that in log forest, individuals tend to be smaller than individuals of the same species in primary forest. And we're still trying to understand why that is. So we don't have the kind of abundance estimates that classically population estimation does deal with. There's another question from Suhail, which is on the slide in the niche overlaps and survival. Which slope is plotted on the y-axis? Survival rate in log forest is what's plotted on the y-axis. So it's just go back to that slide here. So this is only for log forest. So we should have said that. This is only for log forest. There's no primary forest over here at all. So it's survival rates in log forest relative to or correlated with the degree to which niches are overlapping in terms of temperature and humidity between primary and log forest. So the niche overlaps are between primary and log forest, and the survival rate changes are only in log forest. Question from Ashish. Great effort, thank you. Does the State Forest Department have considered your work in incorporating your findings for working plan management, description better managed protected area with sound forestry practices? Yes, the community reserve that I was talking about in terms of working with the Bagun community and potentially going to them with the results and saying, this is what we're finding. Would you consider, this kind of an initiative to protect birds? The Forest Department has had a major role to play in that as well. the forest department, the Mughal community, and researchers, not just us, not just our research group, but a wide group of researchers have been working with the forest department to actually institutionalize these changes. One of the things about Arunachal Pradesh and most of the northeast Indian states is that land management is very decentralized. A lot of the forest land in these locations are about 60% of the land in Arunachal Pradesh actually is managed and traditionally owned by by tribal communities at the village level. And so, if you work with the village council, and the village council is convinced, given all their other requirements, right? They need land for agriculture, they need land for development, they need land for settlements, and so on. But if the village council is convinced, and the forest department is also sort of on board with these initiatives, it is much more possible to initiate measures like habitat protection or management with in keeping with the results that we're finding from research. And so actually a lot of research in terms of not just missetting and population estimation, survival rates, but also in terms of putting out camera traps and saying, hey, look, we've got turbid leopard here, we've got red panda, excuse me, sorry, in these areas, actually has been quite key in convincing tribal communities. But that I should add that that's also because wildlife tourism and bird tourism brings in a lot of money to these areas in terms of employment, in terms of running campsites and so on. So State Forest Department is receptive, it is open. That said, the initiatives would have to be in partnership with tribal communities that own and manage the land.
Okay, I think we're running out of time. Thank you, Umesh, for taking those questions. There are a few questions on Discord, and I would request you to kind of keep the conversation going there. If anyone has more questions, we can keep asking them on that thread on Discord, and Umesh will try his best to answer them through the day. That is the end of our plenary session. And thank you, Umesh, on behalf of all the organizers here. And we will now be moving on to the short talks. Our next speaker is going to be Mr. Sachin Ranade. He is a biologist studying vultures in India for the last 24 years. He has worked on taxa such as birds, insects, reptiles, and spiders. And he is currently serving as the assistant director for the Bombay Natural History Society. Over now to Sachin, who will be telling us about the race to save vultures in India.
Hello, everybody. Myself, Sachin Ranade. I am working in Bombay Natural History Society for last 24 years. And today I will be talking about the race to save the vultures in India. First of all, I would like to acknowledge the RSPB, Royal Society for the Protection of Birds, Government of India, the governments and forest departments from different states in India, and all of you for joining this program. I would also like to thank the organizing committee for this wonderful program. Thank you. So, talking about BNHS, it is the oldest NGO in the wildlife field, and we study the animal ecology, taxonomy, behavior, and we try to conserve the wildlife of India. Indians know about the vultures for a long time, and in Ramayana also it was mentioned, whereas you can see there are a lot of murals or lot of arts and artifacts on birds and bird life. India is a very big country. We have 9 species of vultures. And unfortunately, out of 9, 8 are endangered at different levels. So there are critically endangered species, endangered, and that's why it is really a big race to save these very important scavengers of India. So let us have a look at different species. These are the 4 genera which are represented by 1 species each. So the red-headed vulture, cinereous vulture, bearded vulture, and Egyptian vultures. So all these are represented by 1 species, whereas there is 1 genus that is represented by 5 species, and those are the Himalayan vulture, which is a winter visitor in Indian plains. A similar looking bird, it is a Eurasian griffon, or nowadays it is called as a griffon vulture. And finally, the 3 important species which are found throughout the year in Indian subcontinent. So they are the white rump vulture, also called as oriental white-back vulture, long-billed vulture, also known as Indian vulture, and slender-bill vultures. So slender-bill vulture is nowadays a rarity, and it is restricted only in the northeastern area in India and some Tarai region of Nepal. And here you can see the long-billed vulture. It is a cliff nester mainly found in the mainland India, especially Western India. And both these species were considered as 1 species, and both were considered as a subspecies, but recently they were separated. So talking about vultures, the Indian vultures, especially the old world vultures, are evolved from raptors, and although they have changed their feeding habit, that is, they are dependent now on the dead livestock or dead animals, but they still retain the sharp beak, talons, and they are group loving birds. And that's why they are very much important, because if there are no vultures, then the niche will be taken over by some other scavengers, and which are not very good for the mankind. So what would happen? It will transmit a lot of diseases. It won't effectively utilize the meat, and that will create the imbalance in the ecosystem. So talking about population, what is exactly population is? It is the all the members of that species, and that form a big group, and that is for a layman, it is a population, and it depends on the birth rate, that is addition to the group, and death rate, that is as the individual dies, the population decline. It is also, when we are considering the local population, it is also affected by the movements of the birds. So sometimes there are local travels by the birds, and sometimes there are proper migrations. And this population is dependent on the food availability and the habitat. So here you can see a flock of Himalayan vultures, and you can say that, oh, there are lot of birds. Okay, but it is a comparative term also. In 80s, India was having lot many birds, like 4 crore vultures were there. And Delhi was not just capital of mankind, but it was the capital of vultures also. So many birds you can see in one frame. The photograph was taken by Dr. Gautam Narayan. For the first time, when our team was working in Kevladev National Park, Dr. Vibhu Prakash found that the vulture number was declining. So we had the transects there, and we found that the vulture number was declining, and ultimately there was no whiteback vulture in Kevladev. And that's why this trigger to find out whether this was only found in Kevladev or whether it was a nationwide phenomenon, we carried out nationwide surveys. And everywhere, it was a shocking discovery. Everywhere, there were very few vultures were sighted. Where we earlier had data, we found very few vultures in the protected areas also, in the non-protected areas also. So it was the decline faster than that was recorded for the dodo. So I hope everybody is well aware that dodo was a bird, and it declined due to the hunting. But unfortunately, the vultures in Indian subcontinent also declined drastically, and all these 3 species, especially the white rump vulture, Indian vulture, and slender bill vulture, decline very fast in the late 90s. And the BNHS started working on that, and we found that there was no problem of the food availability. Food was there, habitat was there, and vultures are such a versatile bird that they can nest on even human constructions, houses, all these things. So in 2004, Lindsay Oak and Martin Gilbert found in Pakistan that the diclofenac was the real culprit. And we found the samples in Indian vultures also. The samples were having diclofenac. What happens when the animals, especially the cattle or livestock, are treated with the drug diclofenac? This drug remains in their liver and kidney. And if such animal dies, then the drug remain in the liver and kidney, and such animals are thrown outside. And vultures are scavengers. So when vulture feed on particularly such animals, the diclofenac enter vulture's body and it damage vulture's kidney. The gyps vultures are particularly sensitive for these kind of chemicals. And that's why when the vulture get its kidney damaged permanently, dies a painful death in a week or 10 days. And that's how India has lost is almost 99% of gyps vultures. So when we conveyed this fact to the government, the first step was taken that the birds were considered as a Schedule 1 species. So vultures got Schedule 1 status. We had a big meeting of the stakeholders, and we decided that we should get ban on the veterinary use of diclofenac. That time we know only about diclofenac. Recently, we come to know about other 3 drugs also. Those are the aceclofenac, nimesulide, and ketoprofen. So this drug should be banned, and we should help the bird for breeding. Why particular help was required for the breeding? Because vultures are slow breeding birds. Okay, so we started 4 vulture conservation breeding centers in India, which were completely dedicated for the vulture breeding only. And we were keeping 3 species of vultures in it. So white rump, long-billed, and slender-billed were brought to the captivity, and we were planning to breed those. For the planning, we had a model in our mind in which there should be 25 pairs of each species. They should be breeding, and then only we can release 200 birds of each species in the wild. And that's why as we do not want all eggs in one basket, so we chosen the different locations for these VCBCs. We maintain and manage these centers, and so that there should not be any inbreeding. Every vulture conservation breeding center has a similar blueprint, that is big colony aviaries where the birds will be kept as wild birds. There will be small aviaries like holding aviaries, then quarantine facilities for the birds which are brought from outside or for treatment, and there is a interpretation building where there is a small lab is also located. So holding aviary is a relatively small aviary, but there you can hold about 6 birds together, whereas colony aviary is very big house. It is 100 feet long, 40 feet wide, and 20 feet tall, where the birds can be kept complete in a just like a wild birds. So we do not enter these aviaries frequently, and here the birds are complete living in the wilderness. And here you can see there are nesting ledges. The birds are nesting on these ledges, and all care has been taken that there is not a single pillar inside, so that birds can fly freely. And there is a special net from inside. It is called as net lawn, so that there are least damage if the bird dash against the this kind of net. The vultures are flock-loving birds, and when kept together, they feed together. They like to stay together. They nest together in the loose colonies, and that's how we are working on the captive breeding program. We have a small laboratory as well, and we are breeding the 3 species. In case of Assam VCBC, we are breeding 2 species. So when such birds are in captivity, we have to take all kind of cares. So animal husbandry and veterinary care has to be taken. And you must be wondering that uh, first we saw about the wild vulture populations. Before we start this VCBC, we have to study the vulture population in wild. So now we have to deal with the vulture population in captivity. So it was the ex situ conservation method. For that, we have brought vultures in different ways. So some birds were brought as nestlings and some were brought as adult birds. So in case of nestling, we have to climb very tall trees and bring them to the captivity. For them, a special houses were created and their food regime was also different so that they are not imprinted. And this is how a small chick of white-backed vulture grows, and within 6 months, it can achieve size of an adult bird. But for maturity, we have to wait for next 4 to 5 years. So that's why we need a breeding program, because they are slowly, they get time to mature, and they are breeding with a very slow pace. Trapping is another way to bring vultures to captivity, and we take some chance of rescuing birds. So these rescued birds were also brought for the breeding purpose. Whenever new bird is coming to our center, we are marking it with microchip and rings. We are taking samples, and ultimately, after a certain period, we transfer it to a colony aviary where we are not disturbing the birds. We are monitoring them with the CCTV cameras. So talking about the breeding, so the birds are slow breeding birds. They mature after 5 years. Each year, just one egg is laid. And that's why the breeding rate is very low. And here you can see the vulture A. And in case of vulture, the vulture culture is very much complicated and very much evolved also. Just like today's modern human being, the vultures are very much evolved. The male and female both share the equal duties. They feed to the young, they prepare their own, they prepare the nest, take care of the young. And that's why both male and female look alike. So after say 5 months or 6 months, the birds become mature, and after the bird fledge out, and again, next 4 to 5 years are taken for the maturity. So far, we have bred all the 3 species successfully, and we have started releasing also. And before release, we must study how the bird would disperse. So when the birds are released in nature, we must track them, and for that purpose, we use the PTT. Here you can see a white-backed vulture with a PTT on its back. And when such birds were released, they travel at least 100 kilometers in search of food. And that's why it gives rise to an idea that we should start a vulture safe zone. A safe zone would be an area which is having about 100-kilometer radius. Here you can see the vulture safe zone area in Assam. It has a radius of 100 kilometers. So once we release the birds, it can go in any direction. And that's why we must make that area free from problematic medicines and other dangers for the vultures. So before release, we have to study their natural populations also. And this population is dependent on the food availability. And we have to see if there are vultures, then we have to see their nesting success, and we have to see the food quality also, because the food has caused so much problem in case of vultures. Not exactly food, but the medicines unknowingly used NSAIDs has caused this problem, and that's why the population in India has been dwindled. So to understand the population, we have two very simple methods. One is road transit. Vultures are big-bodied birds, and for that, we have to travel really a long distance, and we have to count the vultures on both sides of the transit. Whereas the other method is absolute count, where we can study and we can count the birds at the feeding ground. In case of road transit, a long road has been chosen, preferably it is a national highway. And in case of protected area, these are the paths which are created by the forest department. Those are followed with slow speed, and we count the birds on both sides. And then we come to know that how much minimum birds could be in particular that area. In case of vulture safe zone, we select the road that will cover most of the area of that particularly safe zone. And then we collect all the data, the age-wise group and their associates, and all the information is uh, is filled up in the data sheets, and that's how we get understanding about the population in that particular area. That was the method road count or road transit. In case of point count, carcass dumps are the best sources, because these are the locations where most of the vultures visit, and you can count them. So here you can see uh, there are uh, there are some tall buildings and short buildings, especially in winter in India. The Himalayan vultures, Cinereus, Eurasian griffon, these birds visit, and that's why the number may rise. And again, it comes down when it is another season like summer. And as vultures are live birds, although we will be augmenting the population, the vultures would be facing some other problems also, especially in Northeast India. There are some poison bait issues. In this case, what happened? The people are irritated due to the stray dogs, and they try to kill the dogs. And that's why they put poison, especially pesticides, on the animal or cattle carcasses. And unfortunately, the vultures cannot differentiate it between the poison and unpoisoned carcasses, and they feed on that, and they are the victims of these unintentional poisoning. There are some problems due to electrocution also, and train heat also. So these are the problems vultures are facing. But the biggest problem was non-steroidal anti-inflammatory drugs. And for that purpose also, we are carrying out surveys, and we are keeping an eye on the pharmacies. And we are finding nowadays that the most of the pharmacies are selling safe drugs. But yet we have to achieve a complete success. In case of population, it is the addition is done only by the breeding, and that's why we are monitoring the breeding colonies of vultures, and we are studying, and we are finding that nowadays the rate of success is increasing. So from this brief discussions, I would like to bring your notice that the vultures are using K type. If there are the smaller birds, smaller rabbit like smaller animals, they breed very fast, increase their number, and again they perish also very fast. But in case of vultures, they are long living and slow breeding, and that's why they have more investment in their offspring. But in case of vulture, if there is a death of adults, just as it is happening due to the unintentional poisoning or NSID problems, then what happens? Even a small number of adults dies, then it may cause a big effect. Especially NSIDs has caused a very big effect. Sometime the population was declining by 22 to 48%. And that's how the vulture population has drastically declined. So we have to worry about that. Currently, the population is at its lowest point, and we are optimistic that if we'll enforce the ban on dangerous NSIDs, it will be really helpful. So I hope that those who are seeing this presentation would be certainly helping us, and the enforcement of ban on these dangerous NSIDs like the diclofenac, aceclofenac, nimesulide, and ketoprofen is very much important in saving our vultures. And as BNHS is having vulture conservation breeding program, we have about 800 birds, and we have started releasing these birds, reintroducing these birds. And from Haryana, West Bengal, and Maharashtra, we are releasing the birds back to the wild. and it is a completely scientific program where the birds are with PTTs or GSM tags. And I hope that we will not be next time just discussing there is a decline in vulture population. There would be some good examples how population of birds can be restored. Thank you very much. If there is any question, kindly ask. Thank you.
Thank you, Sachin, for such an amazing talk. And I think we got a great overview of the vulture declines, causes, and what the current status is. We have one question currently from Paul Pop. He asks, why are drugs like diclofenac not banned for human use, since these are a threat to vultures and might end up in the rivers and eventually on land? So why is it not banned for human use?
Yeah, thanks a lot, and it was a good question. Uh, Vultures are basically feeding on the cattle carcasses, livestock carcasses, or if it is a protected area, then the ungulate carcasses is the main food for vultures. So whatever medicine it is considered, it is going to their body, it is going to be via the livestock bodies. So the drug used for veterinary purposes should be banned as a first step. There was one case that diclofenac was immediately banned in 2006 and it was gazetified in 2008. But the larger vial prepared for the human use was used as a misuse, we should call it misuse in the veterinary field. And that's why in 2015, the larger vials were banned and only 5 ml ampules are allowed for the human use. So as a medicine, it is a very good thing for the mammals or to be precise for human beings. So it is not banned for human being, but its veterinary use is banned. Thank you.
Next question is from Praveen. Do you know if there are no declines of vultures around VSZ, any road transect or point count data that gives us confidence?
Most of the time, the vulture safe zone area is a area where there are small population of vultures. So transacts generally tell us about the vulture population. And those areas where we are going to reintroduce are the areas where generally there are no vultures, or supposed to be, there should not be vultures. And then we are reintroducing the vultures. So yes, in case of vulture safe zone area, there are stability in the vulture population, and we should be working more on that aspect. Yeah, next question, please.
Next one is from Rohit Pansare. Are there any additional challenges introduced to the breeding program due to extensive solar and wind power projects? Are these vulture safe zones being used to define or direct such developmental activities?
Thanks, Rohit. But currently, those areas selected for vulture safe zone don't have either a solar power station or a wind power station. So they are not that much big question right now. But you have very rightly asked, in future, it could be a problem for the vultures if the population or the small population will move towards the location where there are more windmills and such things are there. But otherwise, see, generally, we are dealing with the what are the big challenges, what has caused the 99% collapse in the population. So that's why on the agenda, first is the NSID, second one is the poisoning, and we are working on the newly emerging threats. So yes, we are working on these issues as well. Thank you.
Okay, thank you. We have one question from Preeti about diclofenac. Also applies to cultures and communities that still practice sky burials, where vultures come down and feed on human carcasses. Is that a problem?
Yes, yes.
This question was from Divya Raj.
Okay. Okay. Unfortunately, there are very few vultures or practically no vultures where there are Parsis. Parsi communities are uh, there are nowadays. So especially in the mainland, they are not there, and that's why uh, that is not the question currently addressed. But yes, there are some sky burials in the high Himalayas where especially Buddhist people practice this kind of burial, sky burial. And fortunately, they use very minimal medicines and all that thing. So that's why this problem is not there. And we are not bothered for that. But yes, when vultures will return to the mainland, especially around where and when the Parsis will start again their kind of rituals, then that time we have to make everybody aware that if we are going to practice this kind of burial, then we should not be using the NSAIDs, particularly these four, not only diclofenac, but nimesulide, etoprofen, and aceclofenac as well. This should not be used in the human being.
Thank you so much, Sachin. We're out of time. I would request you to answer any questions that may be on your talk on Discord, and hope you can stick around for the rest of the talks today. Thank you so much.
Sure. Sure. Thank you very much.
Our next speaker is Dr. Shantala Kumar. She is a research associate in the Tiger Cell in the Forest Department of Karnataka. Previously, she has worked on a lion-tail macaque breeding program at the Mysore Zoo and Division of Labor in Termites. But today, Dr. Shantala is going to tell us more about the mass mortality in spot-billed pelicans due to endoparasites. So over to you, Dr. Shantala.
Namaste all. Myself Shantala Kumar present a piece of our work on spot-billed pelican that is chasing behind the mass mortality of spot-billed pelican in South Karnataka. There are 8 species of pelican known globally under the family Pelicanidae. They are widely distributed from North and South America to Australia, including Africa, Europe, and Asia. 3 species of pelicans are known from India, that is Dalmatian pelican, great white pelican, and spot-billed pelican. Dalmatian pelican and great white pelicans are largely known from northwest India, and rarely great white pelicans are sighted in southern regions of the country. Spot-billed pelicans are widely distributed in India. They are resident in some southern lakes, and they are largely known for the local migration. These birds live from 15 years to 25 years, and according to IUCN Red List, they are categorized under near threatened. Their main diet is the fish, and occasionally they feed on amphibian, turtle, crustacean, and insects. Though the media people report on the few deaths of this pelican, during the late 2017 and early 2018, there was a large-scale mortality was reported from Mandya and Mysore district that is largely centered with the Kokrebellur. Hence, we explored further available literature on the mass mortality of the pelicans globally. The review found that the mass mortality was happened in the other species of the pelicans. That is, the mortality was as high as, that is 113 birds were died in pelican. That pelican was died in Florida. And in all the mortality events, the high infection of endoparasites were found. The Contrasecum was the species which found commonly. The Contrasecum was the nematode which comes under the class Chromodoria, order Rhabditidae, family Anisacchidae. This is the only genus in the Anisacchidae family which can infect the terrestrial, marine as well as the freshwater animals. This Contrasecum has the complex life cycle. They need the more than one host to complete their life cycle, that is definitive host and the intermediate host. This prompted us to explore the cause of large-scale mortality of spot-billed pelican in South Karnataka, especially in and around Kokrebellur. Meanwhile, even the forest department was also seeking the solution to manage the situation. Veterinary people were also trying to find out the cause of this mass mortality. We compiled all the mortality reports from Mandya and Mysore district. It found that the Kokrebellur has the more mortality, and few reports were also found from Kukralli Lake, Malavalli Road Lake, Sulekere Lake, and Madarhalli Lake. So the dead birds were also subjected to investigate for the cause of death from the other institutes like Salim Ali Center. The report found that the cause of the death is not due to heavy metals, pesticides, virus, or bacteria. The necropsy of these birds only reported that few parasite worms are found in their digestive tracts. Thus, we investigated for the endoparasites. Hence, previously I told you about the Kokrebellur, which has the large mortality of these birds. Our study also centered with the Kokrebellur. This Kokrebellur is a community reserve which is at Madur Taluk in Mandya district of Karnataka. Kokre means the stalk and the bellu stands for the white color on the top of the tree in the village, hence the name Kokrebellur. For the several centuries, these birds have been present in this place, having the symbiotic relationship with the people live here. These pelicans are nesting in the canopy of the tamarind and ficus tree at the middle of the village. And the people, they celebrate the Deepavali without using the crackers. And they are supporting these birds and they are living with the harmony. And these, on the other hand, these villagers also use the phosphorus and potassium rich manures from the droppings of this bird for their croplands. This village is surrounded by the croplands and the several water bodies. At the middle of the village, it has the large ficus and tamarind tree where these birds are used for the nesting for several centuries. And this, the report which was the large scale mortality has happened in that region. All the dead birds were subjected for the necropsy. So necropsy report has shown that in the preventricular region and the gizzard region of the bird has filled with the adult worms of Chondrocecum, that is mean number of egg or 880 worms in that region. And also the echinostoma is the trematode which was found in the early part of the intestine. It found fishy that since the birds are infected, that is the large infection was happened, so we suspected although these birds are piscivorous and they feed on the fishes, we suspected the fish. So we tested for 6 fishes and 1 fish from the which was fallen from the nest and other 5 from the lakes. So all the 6 fishes had at least 50 to 100 L3 stage worms of Chondrocecum species. Hence, these fishes were present in the water, and also the bird also using that water, and the intermediate species which is also present in the water, we suspected that whether the water is also contaminated with this endoparasite. Hence, we collected the water sample from the 9 lakes which were frequently visited by these pelicans around the Kokkarbellur region. And the samples were 36 samples were collected, that is 4 samples from each lake in the 4 corners, which is 3 meters from the edge. So it found that out of 9 lakes, 7 lakes were detected with the parasite, that is endoparasites. The Contrasecum species found in all the 7 lakes, and the Echinostoma species was found only in the Sulekere Lake. Sulekere Lake was the place where the high endoparasites were found. Hence, we suspected, since the birds which are alive and they are using this contaminated fish as a food and the water to fishing, and even the intermediate host will be there in the water. So we suspected the live birds for the endoparasitic infection. Hence, we collected the 97 droppings from the pelican, and it found that the 51 samples were positive for the Contrasecum species, 67 samples were positive for the Echinostoma species, and 9 were positive for the Ophisthorchis vivirini, out of which 37 were infected with both Contrasecum and the Echinostoma species. 4 samples were infected with all the 3 species. Hence, we summarized, by looking at this present context, we summarized the life cycle of this Echinostoma as well as the Contrasecum species. That is, when the definitive host, that is the pelican or the water birds, shed their eggs through the feces to the water, the egg of the Contrasecum will hatch in the water and develop into L2 stage larva, and this is consumed by the in first intermediate host, that is the copepod. Then there they develop into the L3 stage. When this is ingested by the second intermediate host, that is fish, then when the second intermediate host is directly consumed by the definitive host, the L3 larva will develop into the adult in the intestine of the definitive host. So when they are developed into the adult, the female after the fertilization start laying the egg, and the egg are again shedding through the feces to the water to complete the life cycle. For the Echinostoma, these water birds serves as a definitive host, and when the eggs are shed to the water through the fecal material, the eggs are hatched in the water and they develop into a ciliated miracidium in the water, and this the miracidium will penetrate into the first intermediate host, that is the snail, and first they develop into sporocyst and first and second generation of radii, and then develop into cercariae. This cercariae, after the development, they appears in the second intermediate host, that is the fish, and there they insisted metacercariae. These fish, when consumed by the definitive host, there they develop into adult, and the female start laying the egg, and the egg will be shed through the fecal material to the water to complete the life cycle. Hence, in such condition, when there are few trematode like Echinostoma species and large number of Contrasecum species will become lethal to the bird. During the summer season, over harvest of the fishes by the people decreases the food availability for the pelicans. Less availability of water And fish in the summer probably increases the effort of foraging, lead to the stress. Further, the high endoparasitic infection in fishes add the increase in the infection in the pelicans. Heavy load of these multiple endoparasite in the bird as well as in the landscape are the probable reason for such large-scale mortality of these pelicans. Since the infection involves the water and the fishes, it is very complicated to address. So thus, it requires the stakeholders' involvement. The stakeholders like the forest department, veterinary, irrigation, fishery department, local people and the members of community conservation reserve and research institute, local organization and other involvement is very essential. However, there is no direct solution to treat these endoparasites in this complex system. Acknowledgement to the peoples which are supported to this piece of work. Thank you.
Thank you, Dr. Shantala, for such an interesting talk. And sounds very scary what was happening to the pelicans there. So I had one question before we go on to the questions from the audience. I want to know where are these parasites coming from? Are they there generally in all our water bodies, and there's something that made the birds, like the stress on the birds is what caused this sort of mortality, or what is the risk in other water bodies across, say, Mysore and Bangalore and
Yeah, that is what our question also. See, from where these parasites are actually coming to the birds. So whether it is from the water or whether it is from the fish or which is actually, even the stresses will also increase the number of the parasites. But the thing is, from where it is coming, still it is fish. So,
okay. So we have one question. Do we know anything about the current status of infection in pelicans? Is the mortality continuing? From Preeti, this question.
but the mortality has reduced. So we don't know when it will actually increase, because the summer is the time where this kind of a mortality is whatever we have seen it is happening. So that time only the water level will come down, and also the availability of food also will come down. The number of fish will also come down. The competition will also go high. So that time we suspect that in the summer, so it will go high. So whether it is the answer to the previous question, what she has asked it?
She want to know whether there is like now that the study is done,
No,
is
no.
there still
See, actually, for the initiation, we have interacted with the veterinary people and irrigation, and even with the forest department. So even after our work also, the forest department support us, and they have sent us water sample. So for the periodical assessment, so we have done it and send it back. It is really required. The long-term assessment is required for the what is the current situation and how it has actually gone. So to answer that one, we need a long-term study.
so the latest information, is the mortality continuing? Is the infection still around in the water? I
No,
think no.
after that time it has reduced, and not in that rate. See, whatever we have seen, that is 55 birds in the Kokrebalur itself, it has come down. But the thing is, the mortality rate is there. It is not in that peak. So what it has made in that peak, we don't know.
Okay, thank you. The next question is from Rohit. Are these large-scale mortalities also observed in other fish-eating birds? Could the large size of the birds and their heavy feeding habits make them more vulnerable?
This For that, even this contrasecum species, what I have mentioned is, it is common for all the water birds. See, it will infect all the water birds, and it is possible to infect the birds which are present in that landscape.
Did it infect like birds like cormorants were there?
Yes,
Did it
yes,
affect the? Okay.
yes.
And similar levels of mortality or
It is depending on the immunity level of the bird. See,
okay.
whether it is a susceptible for that endoparasite, or see, endoparasite is actually, it will live in the body of the definitive host. So if the level of the immunity will go down, then only the parasite will actually rule on the definitive host. So that is the reason the mortality will go high. So that's why if the immunity will go down to that bird, then it may have the mortality rate also.
Okay, got it. Got it. Next question is from Praveen. I might have missed it, but have local people reported historical mass deaths in Kokrebellur or anywhere else? Like in the past, has this been documented, or is this the first time?
This even I don't know. Even I
Okay.
never actually heard this type of mortality, but mortality was there. See, few one bird death was there in the Kokrebellur or Kukrelli or in that surrounding area of the water how to say that Ranganthittu area, that bird sanctuaries. So, but the thing is, it is not reported in this high scale mortality.
Okay, thank you, Dr. Shantala. That's how much time we have for questions. There are still some more questions in the Discord channel, and if you have the time, please, if you have time, do go there and try and answer some of these questions. We will. Thank you.
Thank you for the opportunity.
So we will now move on to our next speaker, who is Parveen Sheikh. She is a scientist at BNHS and also an organizer of this conference. She has been studying and monitoring Indian skimmers in the Chambal since 2017, studying their nesting and breeding behavior. So over to Parveen, who is going to talk about monitoring Indian skimmer nest survival, causes of nest failure, and experimenting conservation intervention to improve nest survival.
A very good morning, everyone. I'm Parveen and I work with Bombay Natural History Society. I'll be presenting about our long-term nest monitoring work for Indian skimmer at the National Chambal Sanctuary. So Indian skimmer is listed as endangered on the IUCN Red List, and the population shows a decreasing trend. Less than 3,000 mature individuals have been estimated as per the BirdLife International data, and the species was quite widely distributed in Southeast Asia, but now it's only restricted majorly to 3 countries, Pakistan, India, and Bangladesh. India has more than 90% of the population breeding, whereas Bangladesh does have a large congregation that is observed during the non-breeding season, but there are no nesting records yet. And Pakistan has some less than 30 birds recorded in the past. There are some sporadic records from Nepal, and Myanmar has some very less than 10 birds recorded recently. This species mainly breeds on river, on the sandbars that emerge in summer. They nest in colonies between March and June. And this is how a typical nest of Indian skimmer look like. They nest directly on the ground. They lay their eggs in a small depression that's been created on the sand. And the nesting duration is approximately of 2 months during the peak summer. So the study area that we selected for this monitoring is the National Chambal Sanctuary, which is a protected area on River Chambal of approximately 600 kilometers. And we are working in the 440 kilometers stretch of the river, which is between the confluence of Chambal and Parbati River till the River Chambal confluence with River Yamuna. It's a tri-state sanctuary which runs on the border of Rajasthan, Madhya Pradesh, and Uttar Pradesh. So the methods that we used in understanding, understanding the population as well as the nest survival is that we carried out a total count of the species during February. during in February in the 50 kilometer of stretch. The stretches are divided into 30. The 450 kilometer of the river is divided into 13 stretches, approximately of 40 to 50 kilometers, depending upon the logistic challenges on the river. And all the stretches are covered by boat in 13 days in the month of February. The reason of selecting February is because the species has already colonized by this time, and the colonies don't interact much with the other colonies as they are into the courtship behavior during this time, and it's easy to conduct a total count. Whenever we come across a colony, we count all the birds in the colony and record the location, and we move forward while carrying out our boat surveys. For the nest monitoring, as soon as the egg laying is initiated, we mark the nest. It's a small ice cream stick that we use for marking the nest with a unique number written over it with the date of initiation. Once a nest is marked, every alternate day the nest is visited to understand the hatching success. And once the chicks hatch, the chicks are marked with an aluminum alloy ring with having a unique number to understand the fledging success. And the nesting success is recorded at two stages, hatching success and fledging success. And we also record the causes of hatching and fledging failure to understand what are the main reasons behind the loss, if there is any. So if you see the nest distribution since 2017, we have been recording all the nesting colonies, and the number of colonies range between 20 to 30. If you see the map, you will see that the most of the nesting colonies are located in the midstream and the downstream part. The reason behind, because the river has most of the meanders in the midstream and downstream part. The more the meanders, there will be deposition of the sand and the sandbar will be formed. And there are very few nesting colonies in the upstream part because the habitat is not suitable. There are some rocky stretches as well as rapids in the river in the upstream part. Then we'll come to the hatching and fledging success. I've talked briefly about this thing during the previous bird monitoring symposium. And in 2018 and 19, we monitored around 200 to 250 nests. But if you see the hatching success was 23% and 41% respectively in both the years. And the fledging success was further low. Only 77 chicks could fledge in 2018 and 126 chicks could fledge in 2019, contributing to almost overall nesting success between 10 to 15% in both the years. So we knew that this low nest survival is due to multiple reasons, and the main reasons I'll be talking about are, so if you see that in the river, when the nesting initiates, the sandbar, the drier portion that you see over here, this is the actual sandbar. And later on, as soon as the nesting progresses, this nesting sandbars get connected to the bank because the water keeps on getting low. The river is a highly dammed river, and there are multiple lift irrigations which extract water during the peak summer. So if you see the causes of failure in both these years, the 50% of more than 50% of the eggs were either predated by dogs and jackal. And similarly, that goes for the chicks. There was a high predation pressure by dogs as well as jackal. Similarly, approximately 20 to 30% of the egg as well as chick loss happened due to trampling by cattle that access these islands due to water level receding. So looking at this loss, we just knew that if we take out the predation pressure and the trampling pressure from this nesting colony, the nest survival might improve. And to understand that, we initiated a conservation intervention program known as Gardens of Skimmer. So mainly in this program, what we have done is we have categorized this nesting sandbar into three major categories. The first one being the most vulnerable, depending upon the bank distance to the bank. So the sandbars which are very closer to the bank, and we know that as soon as the hatching is as soon as the incubation is initiated by the nesting birds, this nesting sandbar will get connected to the bank, and there will be high loss. So on this island, we had deployed fencing as well as we have appointed local trained local community members to guard this nesting colony. The second island is the yellow category, which is vulnerable at later stage, where we know that it's not as near to the bank as compared to the red category, but it will get connected as the nesting season progresses, where we have just deployed fencing. And the third category, where there is quite deep water around the sandbar, we have not done any intervention. So in 2022 exactly, we couldn't do much in 2020 and 21 due to the COVID restrictions. So in 2022, we identified 21 nesting colonies and we categorized them. We appointed nest guardians on 4 red category island, and we fenced the 3 yellow category island. The nest monitoring was done as the methods I mentioned before, and we compared the hatching and fledging success on the guarded and fenced island with the non-guarded and non-fenced island to understand how is the nest survival differs between them, to understand the success of this intervention program. So when I say the Nest Guardian program, what exactly I mean is, I'll just run a small video. So if you see, this nesting colony is completely connected to the bank and it's been fenced from one side. So we don't do the fencing throughout the island, because the birds do use some portion of the edges of the island for wetting their belly to come and cool the eggs and chicks. And this is how that the local community members camp on the bank, and they guard this nesting colony, and they don't allow any cattle or predator to reach on this island. When I say fencing, this is the iron mesh fencing that we use for fencing the portion of the islands that connected to the bank to keep the cattle away from reaching the island. So in 2022, we monitored 19 sites having 9 red category, 5 yellow, and 5 green category. And this is how the overall nesting success looked like. So we monitored 380 nests having more than 1,200 eggs, and 329 chicks could fledge out that year. So that was the overall nesting success was more than 27%. Obviously, it's much better than the years where there was no intervention. But obviously, comparing the nesting success of a year with the previous year doesn't give a correct reality of the intervention, because the river is a very dynamic system and the formation of the island changes every year. So what we did is we saw the nest survival on these islands and compared with the control island. So I will just show you the results of the red category island, where there were total 9 islands and where we had protected 4 of them. So if you see, there was a quite significant difference in the hatching as well as fledging between the guarded island, the first 4, and the 5 control island. There was hardly any hatching that happened on the control island, and in fact C3, C4, C5 had a 100% loss. And you can see, I will show you the reasons for this loss that happens to understand how how the Nest Guardian program contributes to the survival of nest. So the causes of hatching failure, if you see on the guarded island, where all of natural causes like sandstorm, desertion, and inundation due to sudden release of water from upstream. Whereas if you see the causes of hatching failure on the islands which was not protected, they were all by predation by jackal, predation by dogs, trampling, and human disturbance. Similarly, if we see the causes of fledging failure also on the guarded island, there was no predation or trampling that happened of chicks, whereas on C1 and C2, there was predation by jackals and predation by dogs. So there was 100% failure that happened on the red category island that were not protected. And obviously inundation was a major problem that year because of the sudden release of water that happened that causes drowning of eggs as well as chicks. Similarly, if we see the yellow category island, which was just fenced, there was no significant difference between the fenced island and the control island. Like there was failure on the fenced island completely, as well as there was survival on the control island in the yellow category. And if you even see the causes, there was predation that was observed on the fenced island. There was trampling which was observed on the fenced island, whereas there was no predation and trampling that happened on the non-fenced island. The reason that there is no significant difference in the nest survival on the fenced and non-fenced island in yellow category was because since there are a lot of sandstorm, the fence did got uprooted. The fence was broken by cattle at multiple places, which gave access to predation and trampling pressure reaching the island. So obviously, we do consider that just fencing is probably not a very appropriate intervention for protecting this nesting colony from predation and trampling. I would just like to show you a glance of the population of skimmers doing on the Chambal. Since the Forest Department has been monitoring the population for a long term, I have just compared the data of 2 decades. So from 2003 till to 2023, the population does show an increasing trend. It is a gradual increase, but since 2020, if you see after the intervention, there's been quite significant increase. In 23, we recorded 740 birds nesting. We have just finished the monitoring of 2024, and the number of birds recorded this year were 840 plus, and it has been a quite productive nesting season, and more than 500 chicks which fledge out on the river this year due to the protection and fencing that we have deployed on certain selected islands. So yeah, I will just conclude by saying that safeguarding by locals as along with fencing is a suitable intervention for the most vulnerable colonies, and only fencing is not reliable. But sudden unplanned release of water is the most detrimental for these birds, and it cannot help in improving the nest survival. Continuing this conservation efforts will definitely help in stabilizing the global population of Indian skimmer, as well as the other sympatric nesting sandbars like black belly tern, little tern, and river tern in the sanctuary. And if you can see that we have been carrying out this Indian skimmer count, which is a citizen science initiative, even the results of skimmer count does show an increasing trend in past couple of years. And we plan to incorporate this intervention in the management plan of the sanctuary so that it can go for a long run. And yes, habitat loss due to large-scale mining in Chambal is a big challenge, not only for conservation of skimmers, but also for other species. I would just like to end my talk by acknowledging all the donors and the Uttar Pradesh and Madhya Pradesh Forest Department who have been quite a support for this project to run for this long run. And obviously, this wouldn't have been possible by many people who have contributed in different ways to this project. So thank you, everyone. And I'll be happy to take questions and discuss about this ongoing project. Thank you so much.
Thank you, Parveen, for that wonderful talk. Made me very optimistic about the survival of the skimmers with just a little bit of protection and lovely videos, I must say. Such a beautiful bird. We have one question from Preeti, who says, very heartening to see the effects of safeguarding. But does the sudden release of water happen often?
So Chambal doesn't have a dry season release plan as such. So what happened in the 22 year is just that the barrage needed some maintenance work, and that's the reason the release happened. According to the policy, they're supposed to inform the forest department before the release, but the release was so minimum and it was not going to threaten any human life, so there was no information that was passed on to all the 3 states. And but it's just that during the peak nesting season, if there is a little bit change in water level, that can drown this, that can completely submerge this sandbar. So that's why it is very important to not make release at a peak nesting season, which is April and May. They can probably do the release gradually from March onwards, so that will not affect this nesting sandbar much.
So Divyaraj has a related question is there any way to kind of convince the various government departments to hold off on the release until the chicks have fledged? Is that something that one can do?
Yeah, so actually, according to the policy of maintaining the minimum flow rate in the river, there is already a Supreme Court law that has been passed, and a minimal flow rate has to be maintained, which is not maintained in Chambal River at the moment. It is below the minimal flow rate. So what happened is just that there is a way of communication from the DFO of all the 3 states of the Forest Department. But before that, there has to be an announcement made that there is a plan to release, which was missing in 2022, which they didn't did, and that's the release this effect happened. We are working with the irrigation department in some capacity building, whether to have some way that the water level is maintained in the river, not only for skimmers, but also the other aquatic fauna like gharial and turtles, because they nest at the same time.
Yeah, so that was Mittal's next question, with like, since the sandbanks are used by gharials and other turtles, do you all collaborate with other organizations to work like to put across like a united front for the conservation of these riverine species?
Yeah, so specifically at Chambal, there's been quite a good collaboration between all the researchers working, especially the gharial team as well as the turtle team. But unfortunately, there is the minimal flow rate that has been already stated. It's based on the work that was done for conservation of gharial that has not looked into the sandbar nesting bird. So I think this is the next thing that we all should do is come up with a minimal flow rate that's going to be helpful for all the species nesting in this river system.
Okay, thank you. Next question is from Karthik. Fascinating work and results. Could you elaborate on the camping by guardians? Did it happen only through the day when cattle and dogs are likely to roam, or did it happen all the time?
Okay, so the places, the 4 nesting island data that we shared, it was 24/7 guarding, because that had a high pressure from jackal, which are mainly, nocturnal presence, which is there. So the guardians do take shift. There are 2 people at night and 2 people at day, and they take shifts of 8 to 10 hours while changing their shift. Yes.
Before we move on, I had one quick question.
Yes.
So what is the difference between, like, are jackals like feeding on these eggs more? Is it wild dog? I mean, is it feral dogs? Because I would wonder whether jackals eating these eggs, is that such a bad thing because they're natural predators in the landscape? Do you want to like affect that as well?
Yes. So the thing is, at certain sites where there is more free-ranging dog presence, we do have predation pressure high or at that sites. But jackal predation is it has increased. So if you see the 22 data, which shows 2018 and 19 data, there was quite a high predation from dogs. Whereas it has changed now, it is more predation by jackals. So mainly if you see in the Chambal region, jackal has become like, this is just an observation that the population has probably quite increased because firstly, there is no major predator for them. Very, very scanty population of leopards around. And especially if you even see that wolf and foxes have kind of disappeared because of the habitat change that has happened. And jackal has probably taken that niche, and they're present like quite everywhere, and morely the garbage disposal by villages is also attracting them, so there's easy food for them. And during the nesting season, especially during the peak hatching season of turtles, which is just before the hatching season of skimmers and black belly terns. So what happened, they are quite attracted on the bank towards the turtle nest, and then later on they are attracted to the sandbars which get connected to the bank. And that is the reason that the jackal predation has been increasing. In fact, especially in the case of gharial, it is observed that they are not just predating on the eggs, but they are also attacking on the hatchlings on the bank of the river and feeding on them. So yeah, that is mainly observation only, but obviously if somebody works on the population, we could understand better about how the population is doing in this region.
Interesting to see that. Thank you for your talk today, Parveen. You can go to Discord. I don't need to explain to you how this
So yeah,
works, but you can go to Discord
yeah.
and answer any more questions there.
Thank you so much. Yeah.
Thank you. We will now be moving on to our next speaker, who is Dr. Anukul Nath. He is an ecologist with a keen interest in addressing conservation challenges through the integration of interdisciplinary approaches. For more than a decade, he has engaged in interdisciplinary research across the Terai region and northeastern states of India. For his PhD, he studied the impact of urbanization on sparrows and will now be presenting about the relationship between sparrows and electromagnetic radiation and what effect electromagnetic radiation has on sparrows. And Yeah, I'll hand it over to Anukul to present his work.
Hello everyone, I'm Anukul from Wildlife Institute of India, and I'm going to briefly talk on the rise and fall of sparrows. As you know, that house sparrow is one of the most common bird species found in the world, and they occupied almost one third of the world. They are distributed almost in all the continents except for Arctic and Antarctic, and this bird literally follows us wherever we go. You can see here the distribution during 1800 is mostly on Europe, Middle East, and India, but now they are almost found most of the part of the world. So a little bit on the species, they are sexually dimorphic. Male house sparrows have a black beak, face, musk, and a chestnut brown head with a gray crown. And there are 12 different subspecies found in the world now, and their diet consists of variety of kitchen scraps, seeds, shoots, insect, and maximum recorded lifespan 12 years. The story of their association with humanity is ancient and so like our own story, only partially known, although there are 7,000 scientific paper been published on sparrows on different aspects. So the clan of the house sparrow, Passer, the genus, appears to have arisen from Middle East. The first intimation of the house sparrow itself is based on two jawbones found in a layer of sediment more than 100,000 years old cave in Israel, and the fossil bird named as Pessa pre-domesticus. With the Neolithic revolution, the species began to live among humans, and they spread to Europe with the spread of agriculture, and then also spreads over Asia. So, how house sparrow conquered the world? And if you see evolutionary, the genome sequence revealed that two genes are mostly responsible for the success of house sparrow. That is collagen 11 alpha, which regulates the craniofacial and skull development. Actually, the skull shape is likely to be an important adaptation to feeding human sources because uh, most of the domesticated crops have tougher sheets, and at the same time, they also has amylase 2 alpha gene, which actually also found previously linked to adaptation of high starch diets in human as well as dogs, and maybe that's why the species found in urban areas around the globe and can digest starchy grains, what human eats, and which helps explained its close bond with people and its love for rice and pizza. But during 1960s and 70s, UK has reported there's a sharp decline of sparrows, and there are more than 60 to 70% of house sparrow has declined in most of the part of Europe. And there are several hypotheses being put forward, starting from reduction in food supply, increasing development activities, which has reduced availability of nest sites, and also the radiation from cell phone tower, electromagnetic radiation. And declaration of Pero is also reported from India, but there is no historical data to compare with. Although citizen sparrow and the state of India bird has a lot of work done in last 10 15 years, and you can see here that overall the sparrow population from 2007 to 18 it's almost stable, but there's a decline in the metros, where the urbanization has reached the peak in Mumbai, Bangalore, uh, Delhi, where house sparrow population continue to decline. I have worked on Guwahati, which is the capital city of Assam, and what is very interesting because Guwahati also have another sympatric sparrow with house sparrow is tree sparrow. So both the species found in Guwahati, and Guwahati is a hill landscape, and it's a hub of Northeast India, and the city is rapidly urbanizing within the last past 20 years. There are 200% increase in built-up area, but still we have a lot of home garden and kitchen gardens in the outskirts of Guwahati, and I have done systematic study in the entire Guwahati on the both species of sparrow. And you can see the dark red color where the probability of house sparrow is very, very high. And these areas are where food resources also available, because these areas are mostly open markets, as called bazaar, and there are a lot of restaurants, and there are a lot of food available in this area. Radio stations are nearby, bus stands, so where the house sparrow is commonly occur. Here you can see the top, it's a house sparrow distribution, and below that it is tree sparrow. Tree sparrows are mostly confined to the residential areas, not in the city centers. And the below picture is depicting the uh, land use land cover and hill landscape of Guwahati, the dark red, the built-up area. So what I found that with the degree of urbanization, the sparrow increases, but when urbanization reaches the peak, the number of sparrow again tends to decline. So it shows that the house sparrow required some level of mid-level of urbanization, but in the area where urbanization reached the peak, there are less greenery, and the top soil is almost paved. There are buildings with glass facades. There are no uh, uh, area to place their nest, where the number of sparrows are less compared to the mid-level urbanized area. Also, it depicts from my study on different habitat types within Guwahati city. It's the commercial area. I have studied residential and building apartment area. Some type houses. These are old houses with much of the area with a green cover. And what I found that there's sharp decline in the tree sparrow populations in the commercial areas, where the house sparrow has adapted a bit. Although house sparrow also found in the area where urbanization at not at the peak, the mid-level urbanization has a mix of commercial and residential areas. Uh, this is the paper which has created a lot of noise. So this is the first paper which comes out that the urban decline of house sparrow may be linked with electromagnetic radiation. I have also studied, and I found that in the Guwahati, the field condition, where I have seen house sparrow and where I have not encountered house sparrow, the electromagnetic radiation not varied much. They are almost same. They are not statistically significant different. And I have also found that electromagnetic radiation also increases with degree of urbanization. As you know, that in city centers, there are a lot of people using mobile phones, there are a lot of towers, and that's why the electromagnetic radiation also high. But at the same time, those areas also are devoid of greeneries. And as I've already shown that house sparrow prefer areas where the degree is the mid-level urbanization and not where the urbanization in peak. So previous studies has not taken all other life history traits, all other variables which are related to life history traits of the sparrow, and therefore they have linked there is a negative association of a house sparrow number with the increasing electromagnetic radiation. But most probably it is the degree of urbanization people has not taken into account. And you can read this paper. It's available and published in Urban Ecosystem 2023, where I've also reviewed all the papers which has found there's uh, a link with electromagnetic radiation and sparrow decline, and I have reviewed and what are the possible outcomes if they have taken other variables into account. That's also explained in that paper. And I also thought that why house sparrow, not other species? What about other species? Are they impacting with increasing electromagnetic radiation? And I found that there is no relation at all, and it all depends on what kind of habitat they are living. If we are biased towards selecting heterogeneous sites within the habitat, and you can see here with increasing electromagnetic radiation there is no spatial no trend with the mean abundance of different species of bird which are also found in the urban areas with house sparrow. I've also put several nest boxes in different parts of Guwahati, and these areas where the electromagnetic radiation are very high, almost more than six. They are city centers, and I have found that the fledglings and the hatchlings has no issue. But I understand this that if the electromagnetic radiation impact may not have an impact in a first generation. But by looking at the level of EMR we had in Guwahati, it's almost like 6 or 7 at the where there is a high electromagnetic radiation. And WHO said that if the electromagnetic radiation goes more than 200 volt per meter, then only it can generate heat and you can have an impact. But in the field condition, this they're very very less that the electromagnetic radiation is so low that no one can and from that heat cannot be generated and there is no chance of an impact in terms of mutation and other harmful uh, uh, cell deficiencies in the sparrows, and therefore it's better to have to aware more people, and we must go for native garden flora, because I have seen even in the city centers the sparrows are found where there are greenery, there is a native greenery. And in a study done in England, it's found that the house sparrows uh, feed their chicks for 10 to 15 days. The diet is mostly of insect food. So they need, although they are granivore, but they need during the early stages of life, they need a lot of proteins. So if we don't have native flora, the insect cannot host there, then their diet will be mostly consists of grains, and there are high mortality rate when they feed more with vegetables and grass seeds in comparison to protein. Protein of larvae, protein food they get from the larvae and insect larvae. So that's why we need to maintain the native garden flora within our uh, home gardens, so that we can save house sparrow for long. And in Guwahati, there are a lot of sparrows there, because I have already mentioned there's a hill landscape and city is growing. It's not, it cannot be compared with other cities like London and Mumbai as well as Delhi. So it depends on what kind of city you are working, and what is the level of urbanization where sparrows can thrive. So if there's a new city coming, there's a high possibility the sparrows increase and they may stabilize or decline in the areas where they're when they reach the peak. And thank you, and that's all from my study.
Thank you, Anukul, for that very much needed study. I can really attest to kind of the study that you have done, because I'm get very tired of telling people that it's probably not electromagnetic radiation that's causing the decline of sparrows in our cities, and it's probably the over urbanization and the loss of natural cover. And it's great to see your study actually prove that to an extent, and it would be nice to kind of put this out there and increase awareness about the real reason for the decline of sparrows in our urban areas. The first comment from Praveen is very nice to hear your results, where you demonstrated that there's no relation to electromagnetic radiation. And he says that this should go into all promotional material, which, yeah, mirroring what some of the points I made. The next question is from Preeti. How did you measure greenness in your work?
For greenness, actually, I have taken several measures. First, I took the species diversity of all native plants within the 30-meter radius. And also, I took, with the help of satellite imagery, 5-meter resolution, I taken out the grass cover and tree cover separately using Google Earth, also manually. And then I made the PCA score out of all these variable, and also includes distance from the nearest green patch. If my random locations are not in the greenness location, so there is a distance from the nearest green patch, and all of these 4 variables I did a PCA score, and then made the final variable as a greenness.
Just a question on the same side. You also had one graph with awareness. How did you get that variable? What was your proxy for awareness?
For awareness.
Yeah, there was one graph where you were showing
Okay, with the
with
greenness and the awareness. Okay, so
greenness and awareness. Yeah.
I also did a questionnaire survey, and there's a Google Sheet form, and I several people on that, and they found, and I asked them to check in their houses they have house sparrows or not. If they have, and what proportion of these are native greenery. So from that, I make out that one. Even in the commercial area where there are greenery with the native greens mainly, so there they can also see the sparrow. So it's a kind of
Okay.
positive correlation with that.
Also, how long have you monitored those nest boxes, which are where there is very high EMR?
I monitored them for 4 years, and
Okay.
almost every year. But one problem that I got there, because I put more than 14 nest boxes in different urban settings, but the sparrows have very high site fidelity, and once they create nest in one place, they cannot directly jump into other place. Although you have put nest box there, so and Guwahati, there are a lot of still places where sparrows can build their nest. So only I think five of my nest boxes where I got the sparrows, but those are all in commercial area where there are no other place to place their nest. So their electromagnetic radiation is more than I think on an average 4 to 5 volt per meter. So I didn't get any. The fledgling success is almost 3 to 4 birds every year in the
Okay.
4 years of study.
And this is probably the same, like the same group that is coming back, right? You said there's high
Yes,
fidelity, so
yes, mostly.
okay, that's good to know. The next question is from Ashish. Being a culture where people believe in respecting animals, they tend to provide food to them. There are people who also provide grains to sparrows in urban landscapes as well. Did you find similar examples in your study area? And also, will it have some impact on their population status?
Yes, same in Assam also. People used to give more kitchen scraps food to sparrows, but due to several reasons, we don't have now open restaurants, and there's a lot of shops earlier which are very open in the markets. So now most of the cities we have like malls, so they don't really help sparrows, because if there are loose markets like bazaar, and there are a lot of food scraps they can get, but people also provide foods, and they are adapting with those food scraps also.
Okay, I don't think there are any more questions. Okay, we'll just give it a second. There are some more questions that seem to be coming in. Yes. So the next one is from Arnav. On a preliminary viewing of the Spanish EMR study that you have showed, it seems that their location in Spain is not as urban as Guwahati. As such, their urbanness might be in the range where sparrows could be increasing, not decreasing. Can we still attribute their results to urbanness?
Uh, It all depends on which part of the city you select for your study. If you select this study in an area with a high urban, with medium urban, so we'll always found that if you only score EMR for your response variable, then we'll always find that the number of sparrows in the very high urban area will be less compared to the mid-level urbanization area. Uh, and same thing has happened for the Spanish sparrow they have studied. You can check the paper I have published in urban ecosystem. It cause correlation doesn't imply causation. So in that I have provided entire set of examples so why they have found this kind of response for EMR. And also I have studied other birds also, not only sparrow. So the results are erratic for almost all species. It all depends what kind of habitat they are. If you don't consider those variables in your model, then you'll always mismatch with your kind of a result you get.
The next question is from Paul. Are feral rock pigeons competing for food with house sparrows? That is because of the biotic homogenization of pigeons.
Yes, they are. In almost all places, what I have seen, uh, the sparrows, rock pigeons are really competing with house sparrow for food grains.
Okay, thank you so much, Anukul, for that amazing talk. there might be more questions coming in on Discord. You can head over there and answer some of the questions there and continue the conversation. Thank you so much.
Thank you. Thank you.
So that brings us to an end of the invited talks. I would like to thank all the speakers for taking out time from their busy schedules to enlighten us about their work on long-term population declines and the probable cause of it. I think we've learned a lot from their talks, and hopefully we can go forward and learn some lessons from their work and probably put it into our work as well, so that we can understand how population declines are occurring, since we already know that population declines are happening. So now the speed talks will, our next thing on the schedule are the speed talks. This will be live from 1:45 to 3:45. They're already up there on Discord. You can start listening to them now. The speakers will be on Discord between 1:45 to 3:45 to answer any questions. You can ask them on the threads over there. And we will be meeting at 3:45 for closing remarks in the lounge channel. So please join us there and continue interacting with the conference on Discord. Thank you so much, and have a good lunch. Thank you.